Today we're going to be talking about inflammation, or the subtle saboteur to overall health. I am Dr. Jasbi. I'm joined by my co-founder, Dr. Alex Mohr. Alex, would you like to give a little introduction on Theriome and who we are and what we do?
As Dr. Jasbi was saying, we are Theriome. Our main goal, our main initiative is really provide deep phenotyping via leveraging systems biology. We're going to go over what systems biology is, but this is a revolutionary kind of perspective, I would say, in medicine. And one of the prime areas that everyone is so focused on right now, and actually is quite insidious and has been elusive in a lot of more, I would say, we would say antiquated, but you could say more fundamental testing, is inflammation. I mean, inflammation is something that can be expressed in any tissue in your body that is having energy turnover. So it could come from even your skin, your muscle tissue, your bone, your gut, for instance. It is all over, and really capturing a good signature of your inflammatory state is, quite correlated to different disease outcomes. So this is highly relevant for anyone that's interested in their health, and I'm very excited to help host this today. I'm going to be taking care of the questions and comments as they roll in. We do have, of course, a 2-hour time slot blocked out. So if you do have some really detailed questions, just go ahead and please wait for the end to ask those. If you have something really, really pressing, go ahead and drop it in the Q&A, and I'll get to it if we can fit it in with Dr. Jasbi's talk. But with that being said, I think it's time to get started.
Yeah, so without further ado, here's just a conceptual overview of everything we plan to discuss today. Inflammation, really the evolutionary underpinnings of inflammation as a process, the purpose of inflammation as a set of biological pathways, and the evolutionary context for the benefits of inflammation, of which there are many. And then the chronic response of inflammation, sort of something that is really ubiquitous among American adults especially, but it is an unmitigated low-grade chronic response to environmental, dietary, and even some causes lying further upstream. And then we'll talk a little bit about the etiology underlying inflammation in disease. And then some warning signs of inflammation, really the obvious ones that I think everyone knows, and some more pervasive and lesser apparent ones that are sort of insidious clues to the etiology of inflammation. We'll also talk about traditional testing, conventional diagnostics and laboratory tests that are designed to assess various parameters related to inflammation, as well as their drawbacks. And then finally, we'll talk about metabolite testing of inflammatory markers and how we have high sensitivity and specificity given the analytical platforms we use, as well as the bioinformatic side of our pipeline. So essentially the data analysis tools we use that allow us to sort of pinpoint inflammation to specific parts of the body, and how this is the next sort of generation of inflammatory testing. So first, we want to talk a little bit about the definition of inflammation. First of all, it's a vital immune response. We have evolved it for a reason as a defense against infections and a preventative aid in injuries. When we are inflamed and we express pro-inflammatory molecules, we are essentially instigating a set of pathways that are known to be effective at removing pathogens and initiating tissue repair. So there's beneficial processes, right? As we mentioned, the activation of immune cells to destroy foreign invaders. We'll talk about which cells are essentially activated during inflammatory response, the chemical signatures that they exude, and how they play into tissue repair and proper immune functioning. We'll also talk about the mechanisms involved in inflammation, the vascular responses, the blood flow to transport immune cells to the site of injury or assault, the cellular responses, such as we'll discuss the role of neutrophils and macrophages, and how they necessarily initiate a inflammatory cascade in order to perform vital functions, such as engulfing pathogens and clearing cellular debris. We'll also talk about the release of chemokines and cytokines extensively, and how they are essentially chemical signals that coordinate and amplify specific immune responses, and what that means in the context of diagnostic testing. So in an evolutionary context, it is a crucial survival mechanism. And the idea of having inflammation is that we should be in a semi-inflamed state in terms of acute response. This chronic low grade inflammation that is really has been implicated as an underlying factor in so many diseases. This is really the long-term consequences of its activation, and really inflammation is a response. And so the key to mitigating inflammation, of course, is to find the source of it, identify the source of inflammation, and correct it with the necessary therapeutics. And that's where we come in with metabolite testing. So a little bit about chronic inflammation. It arises when there's a harmful stimuli that is persistently present, whether that is an irritant or an allergen in the diet, whether that is a systemic pollutant in the environment, whether that is a genetic miscoding that results in malabsorption of a nutrient and subsequent dysregulation in metabolic circuitry. That is a persistent activation of inflammation. And really, it's, again, the chronic inflammation that we see, not so much the acute responses, which are beneficial, but this long-term inflammatory profile that has been implicated in so many of the diseases that are the greatest contributors to human morbidity and mortality, such as Alzheimer's, such as cancer, other neurodegenerative disorders, and of course, cardiovascular disease. So it is triggered by prolonged exposure, and it is a dysfunctional immune response. So this continuous exposure basically results in an immune system that is no longer discriminatory. It has lost all specificity, and it attacks healthy cells as well as diseased tissue alike. And so it creates this sort of unending cascade. And unlike so many immune responses that have sort of feedback mechanisms, inflammation really does not. In fact, so these we'll talk about neutrophils and macrophages and various cell types of immune cells that initiate and propagate these immune responses. And the fact that their product does not feed back and diminish or reduce the rate of their reactions. And really, it is a sort of catch-22. And so the only way to really stop inflammation is to identify its source and really target that as a therapeutic strategy. Most of our therapies for inflammation are really just targeting the sort of reactive proteins and sort of side effects, the molecular side effects of those causes.
Yeah, I think when a lot of people think about inflammation, obviously they know about its associations with diseases like diabetes, as you said, heart disease, but inflammation itself isn't killing you, right?
No, not necessarily. No, it's inflammation that is leading to dysregulated metabolism that's causing cardiovascular disease, plaque deposition. In the context of Alzheimer's, is implicated in amyloid beta generation and tau deposition as well. And so it is causing, it is the sort of ubiquitous underlying etiology that causes various disorders and diseases that we know of and fear. And it's that chronic, again, activation that is the cause. Inflammation by itself as an acute response is perfectly healthy and beneficial.
So not only is it the cause, but it can also be the accelerant, right?
Exactly. And at a certain point, the immune system, if it is exposed to a chronic stimuli for long enough, it will essentially, as we said, lose that specificity and generally start attacking healthy cells as well. And so that is what we're noticing in the profile of these people with chronic inflammation. And so, in fact, you can even be diagnosed with inflammatory diseases. And so that is its own sort of ubiquitous and sort of catch-all umbrella of conditions. And it has its own discrete set of biomarkers as well in the literature. And so really, this isn't anything new. This is really just a returning to an old knowledge that we've had. So of course, itis, when you hear itis, such as arthritis or hepatitis or colitis, etc., signifies an inflamed organ or tissue. And so we have, of course, joint inflammation, liver inflammation, inflammation of the colon, even inflammation of the iris, and stomach lining, etc. So when you hear that itis, you know that it's an inflammatory condition. And that's just a sort of a quick hand, but really the nomenclature shows that these conditions have had, we've known that their etiology lies in inflammation. Even when you consider the historical context of this nomenclature, it really shows that previous generations of physicians and scientists had a very profound understanding of the important role that inflammation plays in various disorders. And we'll get into a few of them. Here are some scientific commentary takes, right? Inflammation drives progression of Alzheimer's. That's in German Center of Neurodegenerative Diseases. Chronic inflammation in midlife linked to brain decline later. Lowering inflammation reduces risk of heart attack. Targeting inflammation could be a way to address multiple facets of metabolic syndrome. MIT News says chronic inflammation drives a lot of cancers. Well, these are sort of, the science commentary of what inflammation is and the deadly nature of inflammation in the context of various disorders. Here we see a review article which aggregated some 200 studies related to Alzheimer's and inflammation and inflammatory markers. What it showed here is that this acute inflammation, this is a quote from the article, acute inflammation in the brain is a well-established defense against infection, toxins, and injury. However, in Alzheimer's disease, disruption of the equilibrium of anti-inflammatory and pro-inflammatory signaling leads to chronic inflammation, neuroinflammation, primarily attributed to activated microglial cells and the release of numerous cytokines. So this is these are the sort of aggregate results of a little bit more than 200 studies. Many studies point to neuroinflammation playing a fundamental role in the neuropathological changes observed in Alzheimer's, specifically the development of amyloid beta, tau protein, the gradual and sometimes unnoticeable shifts between patients with no evidence of disease and those with high pathology controls that don't have symptomology, and then another shift between those in mild cognitive impairment. So there are metabolic biomarkers related to inflammation that can be used to track a patient's progression through high pathology controls, mild cognitive impairment, and then full-blown Alzheimer's that is confirmed post-mortem. Then you also have brain inflammation appears to have a dual role. It's a neuroprotective as an acute phase response, but becoming detrimental when chronic response is mounted. And so these are the general findings that were found to be most ubiquitously significant across the studies that were systematically reviewed and analyzed. So in wound healing, we see, this is on the left, we see wound healing, and on the right, we see invasive tumor growth. So normal tissues have a very organized and highly segregated architecture. And this is how inflammation is involved in cancers. So epithelial cells sit atop this basement that is separated from the vascularized dermis. And when there is a wound or there's a tissue assault, these platelets are activated. They form essentially hemostatic plug, where they basically convert serum fibrinogen to fibrin. And then there's chemotactic factors such as the transforming growth factor beta and platelet-derived growth factor, and these are derived from activated platelets. So they initiate this granulation of tissue. And the activation of fibroblasts, the induction and activation of proteolytic enzymes necessary for basically remodeling this extracellular matrix. So really you have the function here of matrix metalloproteinases, urokinase type plasminogen activators, etc. And on the right, we see an invasive carcinoma, which is far less organized. And this is again due to the inflammation, the neoplasia that's associated with angiogenesis and lymphangiogenesis. It produces this, what we obviously see compared to the left is this chaotic vascular organization of blood vessels and lymphatics, where these neoplastic cells are interacting with other cell types such as mesenchymal cells, hematopoietic cells, and lymphoid cells, and essentially remodeling the extracellular matrix into a cancerous milieu, which is, of course, driven by inflammatory processes. So this is an excellent article published in 2002. And so we've known the implications of inflammation in cancer for quite some time. In cardiovascular disease, we see the same thing. We see insulin resistance and oxidative stress being really the primary mechanisms that are implicated in cardiovascular disease and metabolic syndrome. And those are in turn, of course, rooted in inflammation. So says here, and we copied this verbatim, low-level chronic inflammation state not only has been shown to be clearly associated with the development of type 2 diabetes, but also current data seems to suggest the elevation of certain cytokines are tightly linked to a variety of cardiovascular complications, such as hypertension, hyperlipidemia, heart attack, and stroke. And of course, it says here, the inflammatory activation mediated by inflammasomes through IL-1 beta interleukin compound. Activation may contribute to insulin resistance and TGUM. And in obesity, levels of palmitate and ceramide have been found to be elevated, and these lipids are known to activate inflammasomes. And we'll come back and talk about palmitic acid as well, but this is just to show you that, beside the sort of headlines that we read in magazines and science commentary platforms and blogs, there are a wealth of studies that have substantiated inflammation and its role in cardiovascular disease, metabolic syndromes such as diabetes and Alzheimer's disease, which is itself a form of metabolic syndrome.
I mean, billions, millions, if not billions of dollars have been pumped into looking at these very conditions for decades. So as you're stating, we've mapped this out pretty well. And I think, CVD and going back to Alzheimer's in the previous slide, and certainly metabolic syndrome, this is something that is weighing heavy on our society currently. But there's a time element to it. Right?
Absolutely.
So, when one thinks about cardiovascular disease, for instance, you're thinking about, well, that's really something that's going to be occurring in my, if I'm really unhealthy, my middle ages, but probably, when I'm a bit later in life. But we know that a lot of this damage, particularly in the vasculature, is occurring as early as in your 20s. I mean, even if you're really metabolically compromised in your teens. So I think, the way that we're, with our company trying to provide this essentially diagnostics, right? I think a lot of people are getting more attuned and interested in profiling early for early detection. So, with these types of very prevalent conditions, when do you think it's feasible for someone to start testing for these early signs?
Well, that's the issue with, some things like C-reactive protein. It shows that you're inflamed. It doesn't show how or why. It really is an idiopathic result.
Sure.
If you do other versions, and we'll talk about that are sort of benchmarked against CRP, C-reactive protein, we see that they're incredibly anachronistic. So just like you said, time and temporal result is important. So they are usually variable in their expression. So some people will express this parameter, like an EST, they'll express it, an hour and a half after injury or assault, and somebody else may never express it or express it within days. And so it really isn't temporally associated well with the outcome. And and some of them are not benchmarked. Some of them, for instance, the plasma cytokines and chemokines, as we'll discuss. These are plasma proteins that are usually expressed in levels so low that they're below the dynamic range of the assays meant to detect them. And so they're not expressed in sufficient amounts really to allow the current technologies to reasonably detect them and model them. And so we'll be talking about how inflammation is traditionally monitored and some of the drawbacks with that model. So what are the warning signs of inflammation? So there are these obvious ones that I think everybody really knows. And those are, the obvious signs such as redness, swelling, heat, pain, loss of function. So we know that red areas are usually inflamed because of the increased blood flow. We know that swelling compartmentalizes injury and allows for localized cellular repair and restoration. Heat, again, increased blood flow. These are the results of the inflammatory response, right? First step is to get immune cells to the site of inflammation. And then there's pain, of course, either by irritated nerves or the affected area, and you can have even total loss of function in some cases if it's if the inflammatory juncture is impeding on the transmission of a muscular nerve. You could have people lose the control of their extremities. And so these are very obvious and kind of hard to miss signs of it. What are the lesser apparent symptoms that are still significantly and strongly correlated? Those are aches and pains, fatigue, brain fog. Allergies can reflect a non-specific persistent immune response. Autoimmune diseases, again, you have a immune system that is essentially lost its discriminatory capacity. You have acid reflux, candida in particular, but other microbiome dysbiosis that can cause inflammation. You want to talk a little bit about that?
Absolutely. So this is an area of research that Dr. Jasbi and myself have been actively engaged in for several years now, actually publishing in Febs recently. A lot of the inflammation that is coming from the gut is actually not the bacteria itself. I think that's a common misconception, particularly the idea of what's most people term simplistically as leaky gut syndrome. So this is the idea that you have a degradation in the barrier integrity of your gastrointestinal wall, and certain components are able to come into contact with immune associated tissue and then spur inflammation. This happens principally by components of the bacterial cell wall, which we call endotoxin. Okay, so there are many different types of endotoxin, but basically what happens is these small little components come into contact with macrophages along the border of your gastrointestinal tract. They spur a inflammation cascade that actually interacts quite your liver. I hope we touch on this, the liver really being one of the prime axes of disseminating inflammation in your body. And here at Theriome, I want to make this really clear, and we can touch on this after we're done with this talk, but we're touching on one overall health domain that we profile in our Aristotle test. We have 12 of these. Yeah. And it's often really hard. I don't think we've ever done it in any of our webinars. We don't talk about one in isolation. At some point, we're going to bring up another one. And we do actually have an entire health domain for the liver. And we know that having a healthy liver is really good if you want to handle inflammation. So I will park that for now, but I know we're going to return to this a little bit later on.
Yeah, there's a lot of things that we're going to park, shelf, and come back to, specifically the health domains that we can monitor at Theriome and the really nuanced and tailored approach. So, we have recurrent bladder and yeast infections, as well as psoriasis, eczema, poor sleep or reduced sleep, diminished sleep. These are all indicators of inflammation. The issue is that we all have 3 or 4 of them, and that is roughly the problem. Is that we all, to some extent, suffer from, given the nature of the developed world, given various aspects and processing methods that are in our diet, even hidden and unobserved processing steps that we're not aware of. Environmental toxins, lack of exercise is one of the greatest. Physical inactivity is one of the greatest drivers of systemic inflammation.
There is one environmental factor that I would like to discuss briefly, and that is parasites.
Yeah.
So
okay,
here's just something very simple, and this is not a ubiquitous statement. This is not applying to everybody. But the one thing that modern society has done has helped greatly reduce the burden that we would normally experience living in a pre-industrial society.
Absolutely, yeah. Reduction.
However, on the flip side of that, if you have everything else that's been kind of conditioned and reduced, meaning you're not going to have as robust, basically for simplistic terms, we'll use the term resilience, right? If you don't have a resilient state, meaning you're not able to bounce back to a homeostatic level after some type of perturbation in the system, that's problematic. And if you do get exposure to a parasite in a modern day society, and you are not exercising, you are not eating a good diet, you don't have good liver health, then that can be pretty problematic. That can become an issue.
Yeah, it's a
Just one-
confluence of a few risk factors
Right.
that really make.
And that's an issue that we've discussed recently with plenty of people. And you know, they're not experts in this area, but they read things and you know, they may think they have certain things, but through our profiling, in many cases, we kind of say, well, no, this is not what you have. Actually, you really need to improve the health of your liver. But on the flip side, you know, we do get some rare cases, and we are able to take kind of a step further with our testing, our profiles, that a lot of traditional testing is just not able to handle.
Sure. The question for you in the Q&A, and then I will, while you type that out, the response to that, I'll continue the webinar in the interest of time. So what are some clinical tests for inflammation? You've probably heard of C-Reactive Protein. This is testing active systemic inflammation and can be an early indicator if it is thought to be tested. Significance? Well, it really just sort of discriminates inflammatory from non-inflammatory states, and it can, to some extent, reflect severity. There is a sort of instituted normative range, which is 3 to 10 milligrams per liter in the blood, and it can rise several hundredfold as a result of inflammatory challenges. And that is really the reason why it can reflect severity, because it's the fold change that is correlated with the severity of inflammation. However, it changes rapidly after injury. So when you measure it is really important. It peaks at 48 hours, and then it usually normalizes after resolution. So in the sense of, can it gauge systemic active inflammation and roughly ascertain severity of that inflammation? Yes, it can. However, the testing timeline may not be conducive to a lot of clinical workflows, but it does resolve after inflammation ceases. So it is a sort of good sort of cause and effect node to parameter to test for. Serial ratios may associate with outcomes. So looking at various measurements of CRP per other parameters can be used in critically ill patients to prognosticate outcome. Okay, ESR, or the erythrocyte sedimentation rate. It's an indirect measure of the plasma proteins. It is influenced by various disease states, so it's not really specific to inflammation, actually, and it's has a slower response than CRP. So it's not really effective at measuring acute inflammatory processes. And it has sort of a variable range, and it's dependent on sex and age, because we've seen that it's higher in women and it's increases in older populations. And it's useful for diagnosing certain conditions like bone lesions, osteomyelitis. But in the case of looking at inflammation, it's really a cursory and oblique view of underlying inflammatory processes. You have procalcitonin. So it's released mostly as a result of bacterial infections. It is mostly a tool for analyzing specific sources of inflammation, such as bacterial and viral burden, pancreatitis, tissue damage. Some autoimmune disorders require a procalcitonin test. But its elevated levels are usually indicative of sepsis. And so it's not necessarily at the higher ranges, it's not a good marker of inflammation. It's really a marker of systemic sepsis, which is a far worse state to be in. And it has been used as a therapeutic monitoring biomarker, and it has been FDA approved for the treatment of antibiotic use in respiratory illnesses, essentially monitoring efficacy of treatment and gauging when to cease treatment. And it has some utility in managing treatments and, of course, antibiotic prescriptions. So as a tool to monitor antibiotic use and discourage overuse, procalcitonin has a spot.
So I'm getting a question about CRP, and I think you're going to talk about this in a couple of slides, right?
Yeah.
Okay.
Yeah.
So Sarah, we'll address your question in just a second. We're going to get to CRP in just a moment.
Well, we passed CRP, but we're gonna come back to it and discuss why it's not.
Well, I think she has a specific question. I can just ask it right now. Curious if there is a continuous CRP device / monitor for consumers to use, something like a continuous glucose monitor.
I don't believe there is, no, however, there are companies that are actually working on it. There are companies that are working on a lab-on-chip device, and these are things that can essentially be stuck in you with a microneedle array. They are specific to usually one compound, unless it's a multiplex device, and that's very expensive.
Yeah.
So we are seeing the emergence actually of lab-on-chip devices to measure C-reactive protein. Yeah. Although I don't know of any continuous monitoring either internally or externally that's approved by the FDA. So
use at your own risk.
yeah. Okay, clinical tests for inflammation. We also have plasma, chemokines, and cytokines. As we mentioned, some of these tests are cost prohibitive. We did measure some of them, and these are one of the really more cost prohibitive testing. It's plasma, chemokines, and cytokines. These are these sort of end stage protein expression that we see observed in inflammatory cells, animals, and human models. They're the small proteins. They're really important in coordinating and amplifying inflammatory responses. They direct the cellular movements, specifically immune cells. So cytokines are involved in cell signaling. So they tell these immune cells, neutrophils, macrophages, natural killer cells, antigen presenting cells, where to go and where to aggregate in response to tissue damage. And they include things that you may have heard: CCL2, CXCL8, interleukins, IL-1 and IL-6, tumor necrosis factor, which is, of course, somebody I think most of our attendees have probably heard of. It's TNF alpha. We've heard of these terms before, hopefully, and interferons, and we'll talk about these in detail more. But they're, again, indicating a generalized ongoing inflammatory response. They are not specific to a type of inflammation. They're not specific to disease states. They cannot indicate what is the potential underlying or the most probable underlying cause of inflammation. And they're used in various disorders, just like we discussed. Even autoimmune diseases will typically require chemokine and cytokine panel for differential diagnosis. The C-reactive protein. It can rise in any inflammatory condition. So it's a poor discriminator for any type of disease. To say that you are inflamed is really a non-specific response. And speaking of response, it may not appear until hours after the onset of inflammation. So that may not be, in certain cases, timely enough for diagnosis. And then it has limited predictive value. So when we look at measuring CRP and looking at outcomes or severity in clinical settings, we see that it doesn't have good discriminatory power. What we would usually do is conduct a receiver operating characteristic analysis, or an ROC analysis, and we assess the CRP levels and its predicted outcomes and its ability to discern between severity of disease, whether it's localized, systemic, and predict and classify patients prognostically sort of at clinical intake. So CRP is really sort of a generalized and non-specific marker. When we look at ESRs, these are again influenced by non-inflammatory factors. These can be influenced by really nuanced states such as anemia or normal aging. So it can lead to a high rate of false positives and negatives. It also has very low sensitivity. So a lot of diseases can progress without any sort of significant rise in ESR. And it's also slow to respond. So it can take hours to days to really discern the end of an acute inflammatory response. And what we see here on the right is that it's not necessarily well benchmarked against CRP either. It's really sort of out of step with CRP measurements, and it's complementary, but its use in clinical settings is therefore limited. For calcitonin, it's very cost prohibitive for routine testing. Any one of these tests can run between, if ordered online, you're looking between $359 and $469, which was as of this morning what I found. And so it's a really impractical marker in healthcare settings. The dollar amount spent per parameters returned is not cost effective, and that's really a consideration when designing these tests. You're spending $300 to $400 for one value, and that's really not cost effective, nor does it really lend itself to big data analysis and the insights we hope to get.
So I have a quick question here from an anonymous attendee. So when would you ask for CRP? In what circumstance? And say when would be better to look at ESR? I mean, put yourself in.
Yeah, in a clinician's
Yeah.
shoes. So we're not really clinicians, right? We're scientists, we're PhDs. What we know is what is reported in the literature and what values. But I would imagine in a sort of diagnostic workflow, if differential is being conducted between sort of systemic inflammation related to an environmental trigger, or maybe one that's more autoimmune disorder related. I may order, a CRP, or myself. I mean, I'm not ordering any tests, right? Like as a consumer,
Right.
as a consumer, I would go for, the ESR if I suspect some autoimmune disorders.
And I also think there was some confusion. Can you briefly just touch on the difference? I think there's some confusion here between CRP and high sensitivity CRP. So really that's just coming down to the test, right? So generally when we're looking at CRP, we're referring to HS.
We're not doing non-high sensitivity CRP. And really that is such an outmoded type of test. It's really how it's detected and the dynamic range of that test and how low it can go in its dynamic linear range and still have quantitative capacity. The assay's quantitative capacity is inhibited. And so when we say CRP, we're referring to HS CRP, high sensitivity CRP. And so there's issues here with plasma chemokines and cytokines as well. Really requires sophisticated laboratories. It limits its accessibility, therefore it's also very expensive testing. There is wide inter-individual variability. There's really it's a difficult endeavor to try to ascertain what is a normative versus disease state level. And so it has poor discriminatory capability between the healthy and the disease, because somebody may have a perfectly normal reaction for them. And because it's so idiosyncratic and unique to that individual, it's hard to establish sort of universal cutoff values. And the interpretation of those results, therefore, is largely contextualized and dependent upon longitudinal or serial measurement through time and physician observation interpretation.
And I think something that everyone on this call or on this webinar can attest to. I mean, whether you know it or not, these tests are reliant on a type of assay called enzyme link assays. Okay?
Yeah, ELISA.
And these, I mean, they've been the backbone. They've been the backbone of diagnostics and laboratory work in the medical space for decades. However, they do require a certain volume of blood. And if you go in for routine blood work, how often is it? How many 5 ml tubes are you getting extracted from your body?
Absolutely.
I mean, that's a lot of blood, right?
It's a lot of blood.
Invasive.
It's the inconvenience of it.
Yeah.
Really, for me, I hate sitting there and being poked and prodded a few times.
Well, it's not conducive. It's not conducive to at-home testing.
It's not. No. When we talk about the concept of remote patient monitoring, which is something that we emphasize and enable with all of the testing that we offer at Theriome, both what we currently have on the market and what we have in our R&D pipeline, they are direct-to-consumer point-of-care sampling methods, and they're easy. They're non-invasive, or at most minimally invasive, and really forego the need for vials of blood because of the platform we're using. We're going away from immunofluorescence. We're going away from enzyme-linked immunosorbent assays. We're heralding this new era of mass spectrometry, next generation sequencing, and bringing these research level platforms to the consumer and letting them be clinically relevant. And so we want to talk about metabolomics a little bit here and what it is and why it's important. So the metabolism is nearest to the phenotype. Keep that in mind. So it is the immediate level of your systems biology. So if you've never heard of the metabolome before, it is like the genome. It's like the proteome, the microbiome. Whereas those levels denote the total set of genes and sequences, and the total set of proteins and their complements and isoforms, and the total set of microbiota and archaea and viruses and fungi in and on you, respectively. The metabolome is the total set of metabolites, and metabolomics is the study of those total set of metabolites and their dynamic interactions and influences, both between and within each. And so
I think kind of a good comparison, and I'm sorry for all the simplistic interjections here, but just for a comparison. So like when I'm going to get my yearly checkup at my doctor's office and I'm getting routine blood work, I mean, if I'm really getting the full suite, how many markers am I generally looking at? It's not over 100.
No,
it's not over
gosh,
100.
No.
So I'm saying, I mean, depending on my health plan, right? If I really went full bore and got a very comprehensive panel, we're probably looking, I'm a generally healthy male, so we're probably looking maybe 40 to 60 markers.
Yeah.
When I'm profiling the metabolome from your blood, how many metabolites are we talking about in what we're profiling with our capabilities?
Yeah, our total set of metabolites is 126 metabolites, and this represents 68% of all human biological pathways that we know of.
So I just want to put that in perspective for everyone. The other part of that, and I'm sure you're going to touch on this, but I get excited about this stuff, and I just want to talk about metabolomics with you every time I get a chance, because I'm more of an expert in the microbiome. But one of the fundamental aspects of how systems biology has taken off over the past, I would say probably decade or two, is bioinformatics. Okay, being able to take all of this data and doing something with it, because I can go get 126 metabolites profiled, and just like your physician, I'm given these results. My human brain, I mean, I think we've done a napkin calculation of this before.
Yeah.
Do you remember the number?
It's like 4.5 years that, I mean, given the number of models that we can make with 126 metabolites, the number of combinations being 126 squared, and let's say it takes the physician 5 minutes to build a model, which it's not. I mean, it's going to take them like an hour. But let's say 5
models, just for everyone's reference, what are we talking about when we say a model?
A combination of that greater 126 that allows us to look at something like inflammation and more specifically and sensitively tell where is it coming from and what can we do about it. And so what you mentioned is something that's really important, and it's something that makes Theriome stand out to every single other testing service, including LabCorp and SonarQuest. The idea is that if you go get your values, they are analyzed discretely in a univariate manner. So every variable, like hemoglobin A1C or C-reactive protein, they are analyzed in the context of only itself. And so C-reactive protein can signal how many different conditions, right? Low heme values can signal a myriad of conditions. Low heme plus high or low iron can rule out a few of those conditions. So that is a model. That is a very nascent model, a non-quantitative model that physicians will do in their own heads when analyzing, for instance, heme levels in the context of iron levels. But it's a non-quantitative one. It's a very rough model. It's not validated, and it's not based on the reported science. It is based on their understanding of biology, their experience, and previous outcomes in that interpretation. It's very difficult to go anywhere, and even if you have 40 markers of inflammation assessed, they're reporting them to you one by one, and the interpretation is really up to you. Nobody is running that through a competent bioinformatics pipeline to then make meaning of all that data. And so the metabolome is what we're monitoring. We monitor 126 of them, and of course, as Alex mentioned, we rely heavily on machine learning, artificial intelligence, to model and bring meaning to our collected data. What we use is a platform known as gas chromatography, mass spectrometry. So it is an incredibly powerful platform that is used mostly in research settings. in certain very advanced hospital systems, such as the Mayo Clinic or Johns Hopkins, they may have, they indeed do have, both universities and campuses do have a metabolomics core, whereby they take patient samples that are sourced from their various locations and clinical settings, and they analyze them in one area, and they use that return data. The physicians will use that to inform their treatment. Metabolomics has indeed been incorporated into clinical settings since the late 90s. And it provides us high sensitivity. With a mass spectrometer, we can detect, most of these metabolites are being expressed at the micromolar level. We are detecting them at orders of magnitude even lower than that. We can detect them at the picomolar and some of them even at the femtomolar level. So it's very sensitive. We also have a wide detection range, meaning that we can detect the very sort of early changes in metabolite levels, and we're sensitive enough to detect them at very low levels, so we can sort of make sense of those very meaningful nascent changes that often preface the development of Alzheimer's disease and heart disease and cancers, et cetera. We also have a wide database equity. So many of the tools that we use, including the mass spectral libraries, including the disease enrichment databases, have been built by other researchers working in the field. So we can readily leverage many of the tools we already have in research. We're here in Arizona State University right now, where our office and lab spaces are. And we can leverage so many of the tools that the greater academic community has built. And then when we couple that to gas chromatography to increase the specificity of what we detect, to be sure of metabolite identity. And metabolomics as a science has grown drastically, over the last 20 years. So 24,000 of the roughly 37,000 studies that you can return on a PubMed search of metabolomics study were published in just the last 5 years. And it has already proven to be incredibly beneficial. Since the early 70s to 90s, metabolomics was sort of developing, and in the late 90s, it really began to flourish. And it has facilitated since then numerous discoveries related to diseases, behavioral disorders, drug mechanisms, drug target identification, clinical interventions, patient stratification, and has been a really indispensable tool, promising tool in personalized approaches. So in conjunction with, as Alex mentioned, those multivariate statistics, advanced machine learning algorithms, we can interpret that data and make a cogent report on that individual's phenotype. And so when you come to Theriome to do any sort of health testing, you are getting what we don't call them really reports, we call them studies. These are studies that are performed on an individual, N of 1 studies. You are performing a molecular study of yourself and leveraging our detection platforms, our analytical capabilities, our bioinformatics pipeline to get a sort of molecular fingerprint on yourself and what that means.
So really quickly, I did get a question here that I think is relevant. Becky asked, will the test specify which area of the body is inflamed and why?
Yeah, so we'll get, Rebecca, we're going to get into the metabolites that we use that are the subset of that larger 126 that we profile. We use a subset of 7 of those metabolites, mainly. There's more than 7, but mainly we use 7 of them to ascertain the condition and its etiology. We know that some of these metabolites are only produced in certain quantities, let's say in the liver or in the adipose tissue or in the mitochondria or in the hypothalamus. And because we know their chemical etiologies, we can use their levels, their expressed levels in the blood that we test to pinpoint roughly and relatively accurately the source of that inflammation. And that's what we're going to discuss today. And I'll bring up a content on the slide that shows the tissues that we can pinpoint with these 7 biomarkers, these metabolites that we're going to cover right now. And so here we see taurine. Taurine has been proven to have potent anti-inflammatory properties, and decreased levels are almost always associated with higher systemic inflammation. And so it has general protective effects across many cell types, and they're crucial, especially in the presence of antioxidants and inflammatory cells. So what we see here is sort of a TNF, IL-6 and IL-8 levels, both in articulated adipose tissue and subcutaneous adipose tissue. The control is in a white bar. You can't see them in the first graph because it's so lowly expressed. And here we see these cells are treated with LPS, or lipopolysaccharide, which is a known inflammatory, pro-inflammatory molecule saccharide. And what we see here are significant increases in the cells treated with LPS, significant increases in the cells treated with LPS and taurine. However, they're significantly lower than just LPS. Again, this is true across articulated adipocytes and subcutaneous adipose tissue, and it's true for TNF, IL-6, and IL-8. Now, when we look at IL-1 beta and IL-1 receptor antagonists, we see the same situation between the control. Controls have sort of baseline inflammation when added to the cell control vehicle. And then with addition of LPS, we see a significant rise in this IL-1B in interleukin. And we also see a significant rise in LPS with taurine. However, it is significantly lower still, and significant at the 0.001 level, meaning it has a drastically lower expression as a result of that taurine addition. And we say this, the same thing in the synovial musculature as well. So we see these levels are expressed and mitigated. So we see that when we add the pro-inflammatory molecule, we see a significant rise in all of these pro-inflammatory molecules, these known pro-inflammatory molecules. So it shows proof of concept that the LPS is working. And then we see a significant reduction between LPS and LPS treated with taurine. And we see this expressed again across articulated adipocytes, subcutaneous adipose tissue, and the synovial musculature. Spermine and spermidine. These are potent anti-inflammatory, anti-aging, potent cardioprotective, hepatoprotective molecules. And what you see here is spectra and the associated chemical structure. What we do is we analyze these. How do we quantitate these? Well, we first separate them based on retention time. Given their different chemical structures, they will necessarily have different retention times following chromatography. One will come off of the chromatography column and be detected at, let's say, 9 minutes, and spermidine will come off and be detected at, let's say, 11 minutes. And I think those are their actual retention times, if I'm remembering correctly. And at each retention time that the gas chromatography detects, we scan a range of molecular weights, atomic weights, from 50 to 600 Daltons. And we return identities of those metabolites using their base peaks. So here we have a base peak for spermidine that is between 50 and 100, given its chemical signature. And the spectra for spermine is right just below 120. So by using their specific retention times and mass to charge ratios, we can identify them. And then we actually calculate their quantity by measuring their area under curve. So we look at the spectra, we look at the area that is essentially under that peak, and that allows us to quantitate the levels of these molecules like spermine and spermidine. Third metabolite that is in our panel is, of course, glutathione. We all know how important it is as a crucial role in mitigating oxidative stress, in fighting inflammation, and that low levels typically are associated with very high inflammation. And what we see here on the bottom is, again, the spectra and chemical signature of it. We see with a vehicle and glutathione administration, we see essentially slightly increased production of anti-inflammatory cells. And here we see, these microglia here, we see they're activated, right? And they're really important in mitigating cerebral and central nervous system inflammation. So we see with glutathione addition, we see activation of these microglia. Neutrophils, which are responsible for instigating pro-inflammatory cascades. These are the immune cells that it's their job. They're functioning right. They're being migrated to sites of injury and inducing an inflammatory cascade. When treated with glutathione, we see their expression is essentially completely ceased. And we see significant reduction in expression of, for instance, neutrophils, fluorescence ratios, jellyfish morphology, and the microglial length when we look at glutathione-treated cells. So this is, again, just showing the very potent and I mean, at this point, it's sort of not up for debate. We know the important role of glutathione and its immediate and noticeable effects on cellular physiology. We also have guanosine. It's a nucleoside, so it's a component of RNA, and ubiquitously has been shown to reduce inflammation. And in certain cellular models can reduce, in fact, can rescue up to 60% of the inflammatory profile in cells that are challenged with molecules that are known inflammatory agents. For instance, here we see control and guanosine treated cells. We see no sort of difference there. This DNBS is a dinitrobenzene acid. It is, again, just like LPS, a known pro-inflammatory molecule. So when we dose cells with this compound, we should see rise in pro-inflammatory molecules, which we do. We see that in TNF alpha mRNA levels, IL-1B mRNA levels, and IL-6 mRNA levels. So this is looking at the transcriptomic response to inflammatory signaling, looking at the mRNA that is needed to be translated by ribosomes in order to produce these proteins. And what we see is, of course, with the pro-inflammatory molecule, we see a significant rise in those pro-inflammatory mRNAs. However, when we dose it with guanosine, we see a significant reduction, and that reduction is dose dependent, which is, again, another criteria of causality, whereby usually if it is a mechanistic effect and not merely associative, it should have, it should have a pro-inflammatory and a dose response relationship. So what we see is a reduction at 4 milligrams per kilogram. We also see a significant reduction again at 8 milligrams per kilogram. And that's true for all of the top pro-inflammatory molecules, as well as the ones down here, DCFDA and overall nitrite levels, which are, again, pro-inflammatory molecules, which we haven't discussed yet. There are hundreds of molecules, proteins that are implicated in inflammation, and really they lack the sensitivity and specificity of metabolites, which is where personalized medicine is really interested in taking these inflammatory assays to next. And we see that when we look at Western blots of expression here between NF-kappa beta and beta-actin. So these are, again, pro-inflammatory molecules. Beta-actin is something that we want to see expressed. So with control and guanosine, we see good expression. DNBS reduces that expression. And again, DNBS with 4 milligrams per kilogram and 8 milligrams per kilogram guanosine rescues expression of beta-actin to control levels. And the same is true for NF-kappa beta, just the reverse. Palmitic acid is another metabolite that's in our inflammatory panel. This is an important metabolite that I've actually discovered in my own peer-reviewed literature that's related to breast cancer, Alzheimer's disease. We've even found it in some studies investigating schizophrenia. And elevated levels of this, which are largely diet-derived, are associated with increased inflammation. And it can actually trigger inflammatory cells. So it is itself an inflammatory molecule by triggering an immune response, a non-specific immune response. And it has been actually shown to induce a threefold increase in the production of cytokines by the adipose tissue. So adipose tissue, of course, is a tissue type that is known to be pro-inflammatory. So that's why we have specifically, visceral adiposity. We have very, reproducible studies that have shown significant and strong correlations between increases in visceral adiposity and inflammation. And that is because inflammatories, adipose tissue, fat cells, adipocytes are naturally inflammatory and are really sort of filled with levels of lipids, pro-inflammatory lipids, and pro-inflammatory saturated fatty acids like palmitic acid. And then, of course, we have GABA. GABA, you've probably heard of in the context of neuronal function, and it is an inhibitory neurotransmitter. It is shown to have very high effects in mitigating inflammation in the central nervous system. We see how that is essentially the case with a GABA receptive neuron, decreasing cytokine release. We see decreased phagocytosis and activity by the macrophages. We see more coordinated release of cytokines and chemokines by T cells and natural killer cells. And it's been shown to be potent in reducing even systemic production of cytokines by threefold, again, particularly in those adipose tissues. So when we talk about the metabolites that we can measure, these 7 metabolites we've covered, they are speaking to various tissue localizations, tissues, organs, even subcellular localizations such as the mitochondria or the lysosome. What you're seeing here is a real disease enrichment analysis from real customer data that, of course, is anonymized. But what we can do is take the levels of these 7 metabolites and utilizing our advanced bioinformatics, we can assess the enrichment ratios and the clinical significances of those metabolites, those 7 metabolites, and pinpoint where the most probable cause of inflammation is given these tissue types. And these are only the top 25 that are shown. We can actually significantly monitor more than these top 25, but this is a real output from real collected data. So in this analysis, just by taking a quick look, we can tell that this patient's inflammatory profile is largely driven by hippocampal changes. In addition to the hippocampus, we have metabolites. Those 7 metabolites help us narrow down to the prostate, neuronal inflammation, general nervous tissue inflammation, testes or muscle, adrenal cortex, fibroblasts, the mitochondria. So even cells in subcellular organelles can be typified using levels of these 7 metabolites in concert. Pancreas, brain, skin, erythrocytes, nerve cells, again, at a cellular and tissue level, intestinal inflammation. There's a lot of research being done currently in ER inflammation or inflammation of the endoplasmic reticulum, as it really disrupts cellular transport and kinetics and can result in rapid cell death. And so inflammation is really being studied in the context of the endoplasmic reticulum. Of course, kidney, bladder, spleen, placenta. These are the top 25. But it just gives you an idea that, given the known expression levels of these 7 metabolites, there are known implications in various studies that have shown their etiologies to be associated with either specific diagnostic health outcomes or disease states of these tissue types. We can use our modeling techniques, essentially our bioinformatics, to assess a clearer picture and define the inflammatory profile at these levels, where, again, traditional measures of inflammation, such as CRP, such as chemokines and cytokines, really just point to general inflammation and don't have the scientific backing to be able to sort of conduct this very targeted analysis of the etiology of that inflammation. So this is our first systems biology test that we're offering at Theriome. And this is, again, one of many tests. We plan to build out an entire ecosystem of systems biology testing, going from the metabolome all the way up through the genome. We have our ILIA test, which is a gut microbiome test being released in the second quarter of 24. And so we hope that you'll join us for webinars related to the gut dysbiosis, gut resilience, and all the topics we have planned. But currently we're testing metabolites, and this is the process of the Aristotle test. Sample collection is very easy. You get a direct-to-consumer at-home kit shipped to you. That is what it involves is essentially a dry blood spot analysis that is conducted within 20 minutes and sent back to our central lab here in downtown Phoenix for processing. And that's sent back in a prepaid envelope. So this is a very minimally invasive, patient-centric method of sample collection that is meant to be approachable and still has been proven to give us the data fidelity and the analytical performance in laboratory to be confident in the results. So the proprietary filter paper on which you collect your dry blood spot sample has been shown in reproducibility studies to maintain metabolite integrity for up to 3 months. Really, roughly median time was 2.5 months. analysis. And so it's far more than enough time to send it back to a laboratory for analysis. And how do we analyze that? Again, we reconstitute your sample, we tag it with various internal standards that allow us to quantitate those peaks that we see later that each correspond to metabolite levels. And what we do is we first dissolve the sample matrix, which is the proprietary filter paper. We precipitate proteins. We then separate the matrix, which is the paper, from the aqueous metabolite layer. We then sonicate further to ensure that the metabolites are fully removed from protein conjugates as well as the filter paper. And then we extract that layer of purified aqueous metabolites. It undergoes derivation. Essentially, it's a means of lowering the boiling the boiling point of these metabolites so that they're increasingly volatilized and are subjected to GCMS analysis. And that is how we are able to do a very high throughput analysis and monitor 126 metabolites that, again, are letting us monitor 68% of all known human pathways, which is by far the most advanced metabolic test currently offered, full stop.
And I want to reinforce the term you used, high throughput, because we do have high throughput here at our facility. And just a great example, I mean, our texts are great. What's our turnaround time normally?
Once we get the test and it's marked as received and customers receive the sample receipt email, turnaround time is 1 week.
And again, I have to use the analog to this going to your physician. I mean, assuming you might need an appointment, it could be months before you actually get interpretation, not just the results in your portal, but interpretation.
Absolutely.
We're talking about you ordering the test, taking it, sending it back into us, getting it within, as you said, 10 days.
Yeah, it's really 2 to 4 days to get it. It's 2 to 4 days to get it back. And then we check our inbox, we mark them as received, and within 7 days of that email, they can be sure that we've analyzed the data, we've checked it, or we have ensured quality, analyzed it again, and delivered that report using our secure end-to-end encrypted. That's great. And so this is again just a quick look at the inside of a mass spectrometer and how it works. So you yourself are the source of the metabolites. The metabolites, because of where they sit in the hierarchy of systems biology, right? Genes are transcribed to RNA. RNA is translated to proteins. Proteins interact with other proteins, the microbiome, and metabolites. And metabolites are ultimately the end stage of expression. They're also nearest to the phenotype. They are nearest to the environment. They are necessarily, metabolism is necessarily at that nexus point between the genome and the environment. It is the interaction point. It is the cross-section of gene-environment interactions, which of course, gene-environment interactions are the cause of almost all human diseases and disorders. Human diseases and disorders are not genetic. They are not purely due to genetics. Most, the big ones, the ones that we would say again are the greatest contributors to human morbidity and mortality, those are cardiovascular diseases, neurodegenerative diseases, cancers, metabolic syndrome, et cetera. These are not genetic diseases in the sense that one or two or even a subset of genes can predict or necessarily guarantees that outcome. They are a gene environment result of that gene environment interaction, that give and take. And so it doesn't make sense to test genomics for most health conditions. Genomics is a marker of potential expression. Metabolomics is necessarily a dynamic snapshot of cellular physiology at the moment, and is therefore incredibly sensitive to various conditions and lends itself well to clinical assessment, diagnostic testing. So you are the metabolite source. You, your genes, your environment. We collect that, we again grind, extract, derivatize, we do first order separation with chromatography, and then we get to this mass spectra. So at every, you see a chromatogram, and then you see that inset of the chromatogram is sort of blown up. At every time point, we can extract the mass spectra, and that is how we ascertain levels of these 126 metabolites. Here you see an abridged list of these 126 metabolites. They're abridged, as you can see, because there's just not enough space here on the page. But you get a sense of things that we cover, and probably some of them are immediately recognizable to you. We did discuss, for instance, palmitic acid, number 49.
I want to pause you there really quick,
Yeah.
because I got a question related to palmitic acid. I was trying to figure out where to drop this in. So, good question. Doesn't palmitic acid promote the formation of glutathione in the liver?
Yeah, palmitic acid does a lot of very beneficial things. And so palmitic acid, we need some, we don't, and that some usually has a much narrower range than the full changes we see palmitic acid typically expressed in customer data. So yes, you do need palmitic acid, not just for glutathione, you need palmitic acid for myelination of nerve cells. You need palmitic acid for generation of various intermediaries of the TCA cycle. Lots of fatty acids are used in those regards,
Sure.
especially myelination. Low levels of chronically low levels of palmitic acid are implicated in persistent neuropathy. So yes, absolutely. And for all of these metabolites, really you need to know what is the low range, what is the high range, and where are you? And that's something that you will get with every single one of our tests. You will get an output that says, here's the metabolite, here is the name of it, here is your value. Here's what a low relative abundance is and what a high relative abundance is, as reported in the literature. So these are the cutoff values that the literature says, because many of these metabolites are not integrated quite yet into traditional medicine and diagnostics. They require people like us to sort of build them into models, assess their classification, publish on them, prove them, validate them, statistically, both internally and externally. And so we have levels that we can say, this is where the science says, but below this level, you're at risk of these, clinical manifestations, and above this level, you're at risk of clinical manifestations. So with palmitic acid, absolutely, most of these metabolites, too little is bad, too much is bad, and that's the case for almost all of them.
But I think this is a good point, because again, we don't want to put ourselves. It's so easy to do with our human brain. We just work like this. We want a binary with a single metabolite.
Yeah.
But the model is what's providing the context. We need to know what's going on overall. I mean, for instance, a lot of the work that was done looking at palmitic acid actually spurring glutathione production was done in vitro.
Yeah.
Okay. Very, very isolated. Now, what happens if you're talking about someone that's in the late stage of life, has hyperinsulinemia, is obese, and is eating a lot of carbohydrates and producing a lot of palmitic acid? It might be making some glutathione, but it's certainly not going to be enough to really buffer against all those other noxious agents going on in the body. So modeling is important, and kind of the overall metabolic profile that we're capturing here, putting that into context, like palmitic acid into context with everything else. We just need to be aware of context.
Great. Yeah, you're absolutely right. It's really the modeling, and a lot of people may be able to go somewhere and get some of these metabolites sort of pieced together in a test, but then where's the bioinformatics? Where's the meaning? Where do you make sense of all of this data? And so we, as we mentioned before, we said we're going to shelf this and come back. We monitor 12 different health domains for which we have trained models to assess levels of metabolites that are known to impair or improve these health domains. So in every domain you see is a collection of metabolites, just like the inflammatory score that we discussed. And they're there because they're proven, relevant, and quantifiable evidence in the peer-reviewed literature, which we've mined, we have modeled and validated internally, such that we know that. What do we mean by validated? It's a statistical term, meaning that we're sure that the model isn't overtrained on our data and has room to accommodate new cases, which would of course be our customers. So in addition to the inflammatory profile, we have markers that are validated to infer to aging, nutrition, mitochondrial health, cardiovascular health, reproductive health, everything you see here, environmental toxicity, mutational load. You may have seen some of our webinars on the different domains previously. But this is let's take a step back and say, well, we have 126 metabolites. We have a wealth of literature that shows us qualitatively and quantitatively how and what they're implicated in. And we have the ability to construct sophisticated algorithms that can assess a patient's profile along those parameters and return a score 1 to 10. So this is a very quick overview that we give in every report is a bar graph depiction of the customers. And then we also have a disease enrichment analysis. So as I mentioned, we have excellent equity in metabolomics, database equity. For decades, there has been, so many, scientists, thinkers, scholars, researchers that have added to databases and our collective knowledge of metabolomics, different metabolites and their implications of different diseases. So what you're seeing here is a disease enrichment report that is generated given an open source platform we use, known as the Human Metabolome Database. And what we've done is we've characterized a disease signature or a health state, such as acute signature, such as acute seizures or metabolites affected by exercise. And we've given a total number of related metabolites. So of our 126, how many metabolites do we use to monitor acute seizures or schizophrenia or histidynemia or early markers of myocardial injury or heart failure? And we can do this for 344 diseases that are reported in human blood. Here you see those conditions for a real customer that were significant matches. And by significant, we don't mean has a high risk percentage. Some of them, as you see, have very low risk percentages, like 21.05% for diabetes, non-insulin dependent. The reason we say significant is it's a statistical parameter that ensures that we have a type 1 error rate of less than 5%. That type 1 error is the percent chance that what we are saying is a real match is not a real match. And so we have statistical and research level tools that we're applying to single sample profiling. So we corroborate the customer's metabolic profile across these 126 metabolites with the known disease signatures and health states in that database. So there's 344 of them, and then we report the ones where there was a significant match. So the match is not due to chance, or at least the chance of that is less than 5%. And so we can give a very sort of short list of conditions. And this is really an excellent analysis for customers who have persistent and vague symptoms that they've tried in earnest to diagnose and haven't been able to.
So actually, I think this offers a great point to ask a little bit more complex question from Rebecca. So thank you for being patient, Rebecca. Basically, she's been suffering from arthritis in her leg, and it's really been debilitating to her. She's been diagnosed with inflammatory arthritis caused by an unknown immune disease. It seems that prescription non-steroidal anti-inflammatories are really helping her, and if she doesn't take them, it really kind of cripples her. Can you give Rebecca any insight, or maybe you want to handle this one offline?
Yeah. Let's handle this one offline. Rebecca, it definitely can give you insight into where this inflammation is coming from. As we showed in a previous slide here, let me see if I can bring it up. These are just the top 25 areas that our inflammatory markers can address. There are close to 80 of them, I would say, maybe high 80s that are indexed in this database. And so by using metabolite levels of these 7 biomarkers, these inflammatory biomarkers that we've covered today, these are just the top 25 that we can pinpoint. So if your inflammation is in these 25 or in the greater 88, if there's a significant match, we can say, well, most likely, let's say this is the case, that your inflammatory profile is due to changes in the hippocampus. And we can give both an absolute and a relative assessment of that inflammation, again, relative to other sites that are profiled in our database. So yes, the short answer is yes, we can, but please connect with us with your specific concerns. You can also book a 30-minute slot to chat with me during my weekly office hours. Those are on our website. So if you want to get a little bit more information on this and how it relates to your specific health needs, by all means, go to our website, therio.me. You can schedule a time during my office hours, and I'd be pleased to chat with you more one-on-one. So this is just an idea. In this panel, in our disease enrichment panel, we have various cancers, we have various neurodegenerative diseases, we have, of course, you've seen heart failure and early markers of myocardial injury on there. Many disorders related to aberrant metabolism, digestion, absorption, and that really informs our next step. Alex, do you want to discuss our next step? This is
Sure.
really your domain here.
So I think this is really game changing in terms of how we kind of, in my opinion, how we're going to ultimately view medicine and interventions and treatments. So traditionally, when you go to your physician, you're getting blood, you're getting blood work, you're getting some type of diagnostic work. They're comparing your data to benchmark standards, references of a population, and generally it's benchmarked against similar profile of your age, your gender, etc. However, we're all very different, right? We're all giving off a different signature of what true health for us is. So what we're able to do, and this is actually something that did not originate at all in medicine.
No, this is very new in the concept of medicine. This is the bridge between bench and bedside. And want to give a little bit of
this is the bridge
provenance.
This is the bridge from aerospace manufacturing to now what we like to call precision medicine, right? But basically, digital twinning is a practice that's used, initially was used in NASA. They're engineering these different parts. Obviously, if you think about how expensive it would be to trial all these individual parts for different stimuli that a space shuttle might experience going into orbit, that's going to get very expensive. If we're able to kind of troubleshoot in silico, meaning in your computer, okay, hence you see there the chip there in silico modeling, we can get a very good idea of, if we're looking at a aerospace part, in silico, we're giving it all these different types of stresses that it might experience. We can get a very good idea of, what we need to change, or what the state of the material is, and how we need to proceed with manufacturing. Now we could actually take this idea and bring it over to the human body. If we're profiling your cellular physiology, your metabolism, basically, with our Aristotle test for the metabolomic profile that we have, we can get a really good idea of what you're going to best react to and what you're not going to react to well. So this is the idea of the digital twin. We're forming you in silico with our modeling. And from this, we can give really, really good recommendations, extremely granular recommendations based on your physiology, not some reference stamp, okay? So from this, you see two trajectories in that line graph, the green being like an optimized you. Like, what if our digital twin, we took them on a health journey and they did every single thing right over, I don't know, a year, right? We can see here they're really improving their health score. Now, humans, we deal with real life, so it's not probable that we're going to be able to do the same type of rigor that a in silico model would be able to. However, this can give us a good indication of, to bridge that gap from where we currently are to be at our most optimal state, what we need to accomplish. And this is the power of, I think, one of the main pillars of our bioinformatic pipeline, what we're able to accomplish. Going back to thinking about individual metabolites, this is really going one step beyond just basic pattern recognition and forming metabolism or metabolic pathways. This is going into an individualized, a personal approach to your health, which I think is we can't talk about it enough, but I think for the sake of time, I'll probably wrap it up and end it there.
Yeah. So essentially what Alex said, this is coming from the aerospace and advanced manufacturing sector, whereby parts for various very expensive things like the fuselage of a spacecraft would be modeled, tested, re-engineered, all in silico. And so therefore, time to iteration is significantly less than if you were to produce and fabricate it. So you can imagine how much time and money is saved, and we do the same thing. We have over 1,300 different interventions that are banked in our own Theriome database, and we have gone through scientific literature, and we have mined results, we have mined effects sizes, we have mined clinical significances, directionality of change, both from reported statistics as well as raw data that is often accompanying published research nowadays in online depositories. We would download that data and model it and sort of simulate on the digital twin of that individual their predicted reactions and the predicted effects of things such as prescription medication, over-the-counter supplementation, dietary changes, exercise routine changes, all from various studies, whether epidemiological, observational, cross-sectional, randomized control trials of these interventions. And what we do is we simulate each one of those interventions. Let's say it's the Mediterranean diet. We simulate that on the digital version of you 1,000 times. Each one of these interventions is simulated 1,000 times, and the effects are averaged with a degree of randomness built in. And then we pick among those top interventions which were predicted in silico to give the real individual the greatest response. This is digital twinning. This is personalized care. You will see this utilized more and more, especially as companies try to get a user interface attached to it and make it really usable in clinical settings. This is how we are going to bridge the knowledge that is accrued in the scientific world, in our world, with what the physician, hospitalist, or general healthcare practitioner can execute and implement on their patients, and translating the results of studies to protocols for an individual. So I can talk more about how innovative this is, or we can show you what a personalized protocol really looks like. And this is a personalized protocol that was generated for a real customer. So given her inflammatory profile of 6, so she's in the normative range, but there's obvious room for improvement. We asked her to eat specific foods: salmon, mackerel, sardines, chia seeds, flax seeds, and walnuts. We asked for 2 servings of fatty fish per week, and for plant sources, a tablespoon of chia or flax seeds daily. Preparation, again, to maintain nutrient integrity and subsequent absorption: baking, grilling, steaming. Adding this to salads or smoothies, just really sort of recommendations that have been shown, at least even in the literature, to be important. There are randomized controlled trials where nutraceutical smoothies are given to one group and a control smoothie is given to the other. So these are all gleaned from the literature. Omega-3 supplements: combined EPA DHA intake with a balanced 1-to-1 ratio with 1,000 milligrams per day. This is based on high arachidonic acid and eicosapentaenoic acid ratio. So these are 2 metabolites that we didn't discuss, but are calculated and used in our recommendations for the inflammatory profile. There's 7 metabolites that we discussed, and we use many more of that, the greater 126, to bring you the granularity of this protocol. Turmeric supplementation: we're going to start with 500 milligrams of curcumin with piperine daily, taken with meals to enhance absorption. The purpose is, of course, curcumin has been shown to be a potent reducer of inflammation. The metabolomic profile suggests that there's issues with gut absorption. So we're then recommending to her liposomal curcumin formulations for enhanced bioavailability. And then we want her to follow this for 6 months and then retest after the 6 months. So the algorithm is also providing a retest timeline. So it's not just here, year 6. These are the metabolites we monitored. These are the levels. These are the disease states that you're associated with. But the idea now here is to say, for every health domain, provide a personalized protocol, scientific reasoning, and retest timelines based on when we expect to see changes of those interventions reflected subsequently in the metabolomic profile of that individual. We also added anti-inflammatory strategies. We saw a signature that was related to poor hydration. Poor hydration is one of the leading risk factors for systemic inflammation. Regular exercise and stress management techniques were recommended, as this individual did not exercise. We mentioned earlier, physical activity is one of the primary drivers of inflammation. So for her, we recommended at least 10 glasses of water a day, or 2.5 liters. This was calculated based on her thermogenic metabolite levels, her reported body weight, the climate that we assessed via her zip code that was reported, and her self-reported activity level. We can tailor very customized hydration regimens for individuals as well. And then, of course, we wanted her to maintain a diet that was rich in these fruits, these vegetables, and lean proteins. We specified specifically 85 to 113 grams of lean protein per meal. This was for comprehensive anti-inflammatory support to sort of complement her protein profile and the metabolites related to protein ingestion. And then we added that for her, there was a particular benefit of berries, leafy greens, green peppers, and beets, given various micronutrient deficiencies that we detected in her nutritional score that were also predicted to be driving her inflammatory profile. And so everything is related. I mean, we talk about these domains as if they're silos, and they're absolutely not. Your mutational load is affecting your environmental toxicity, is affecting your nutritional status, is affecting your inflammatory profile, is affecting your cardiac score and your liver profile. These are all sort of related conditions. It is only for sort of easy digestion that we do break them up into these generalized silos. Each one of those silos within them has specific signatures. Again, we saw how we can use our data to inform and pinpoint the specific source and pathogenesis of inflammation. So inflammatory score might be a very generalized concept, but our modeling and our comprehensive data collection enables us to pinpoint the source as well. And so this is what you'll find on our website. That's therio.me. We do have an affiliate website, so if you go there and the offerings and the prices are not quite the same, that is not our website. We have our own therio.me website. These are the different tiers that we have available for testing to, really sort of give you whatever tools you feel like you need most. We work with physicians that are on our practitioner platform, and they use this in their own practice. But you can access the health score report. With all of these different classes, by the way, come with your metabolite levels. So every test comes with, here's all 126 metabolites, their levels, the related, the known ranges for high and low, a general description of that metabolite, as well as implications of high and low levels. And everything is cited in that report and encourages education and follow-up on the customer side. But for $199 or 25% off, we offer our health score report, which is just your, your levels of 126 metabolites and your scores across 12 domains. The personalized protocol is another $199 if you want to add that on. That undergoes the digital twinning experiment. So each one of our interventions is simulated 1,000 times on your digital metabolic profile, and a protocol like the one we reviewed is generated for every single health domain. For $299, you can get access to the disease signature report, which is, again, the enrichments of those 344 disease signatures, and we present the ones that were significantly matched. Or you can get the full report, which covers everything that we went through today, the 12 health scores, disease enrichment, and the personal protocol that is a product of the digital twinning experiments. We also offer a full report and a consult. These are for patients who feel like they may have extra questions, may need further guidance. We have seen that compliance with the protocol is much better after doing a consult with one of the members of our science and medicine team. You will meet with either a PhD or an MD. You will not meet with anybody that's not qualified in the conductance or practice of personalized medicine. And that's $750, which is the full report and a consult where you get 30 minutes to sit down with a dedicated member, again, a PhD or MD member of our team, to discuss any questions, get finer granular specifics on your personalized protocol, review retesting timelines, etc. That time is yours for you to do with as you please. And it's all on our convenient telehealth scheduling and meeting platform. So very quickly, that is our presentation. We'll now get to more questions that you may have. This is our beautiful building that we're actually in right now. We're on the 3rd floor here. Our laboratory is on the 3rd floor here. Come visit us anytime if you're in downtown Phoenix, and we're happy to schedule a lab tour for you. If you're attending today's webinar, you can use code inflammation to receive 10% off of all of our current testing services. Non-subscription testing services, those are already discounted 25%, but the code expires December 10th. If you have any questions, we'll take them now. If you have any questions that come up in your mind after this webinar, please email us at info@therio.me or contact our 1844 number. That's the building, the Wexford Science and Technology building we're in, and our mailing address. So without further ado, I know we've taken up too much time. But let's get to some of the more pressing questions.
So I think the first question is that we need to get to is from Carol, and she's so excited. She actually, I think she took this during the talk, the test.
Oh, did she?
She has a test, but she's just a little concerned. She made the mistake of touching the card. She wants to know if it's okay or she needs to redo the test before she sends it to us for us to analyze.
Yeah, great question. Did she touch the card on both demarcations?
She says she touched the sample area, but didn't indicate if it was on both sample areas. Let's see.
She says, I don't remember.
Don't remember.
Okay, well, since the sample's already taken, you know, what we would usually do is we get both samples into our lab. We sample one, we punch one of the blood spot cards and process that. That's actually all the volume we really need, 20 microliters roughly. And the other dry blood spot card is, you get 2 with your kit, and the other one is stored at -80. And we keep that only until we get the data off the mass spec and we can do a quality control check on it and ensure its fidelity. And once we, ensure that, these are, most metabolites are detected with a signal to noise ratio above 3, then we discarded the second dry blood spot sample. In her case, since she's not certain if she touched one or both or which one, I would still recommend sending it in. If there is an issue in the data, then we will be able to see that. We'll actually see a change, a really drastic change in lipid metabolism. We'll then be able to sample the second one. If we still see that same aberration, then we can send you out another kit. But I still think if you've collected it, it's worth sending in. We've had this issue before. I know our instructions say to, at as much as possible, prevent directly touching the finger tip. However, if it is the finger that you've disinfected, the chance of cross-contamination is actually very low. So we will have to see. Worst case scenario, we send you another test kit, but it's highly probable that the sample's analytical validity is fine, or at least one of them is.
All right, thanks for the question, Carol. Good question. Becky asks, I'm supplement sensitive. She gets headaches, bloating, even insomnia. Is that addressed in the test?
Yes, absolutely. So we have the ability to say to individuals if you should get a certain micronutrient, vitamin, or mineral from the diet or from a supplementary synthetic source, given certain profiles, metabolites related to barrier function, enterocyte activity. We can recommend whether, for instance, the omega-3s should come from 150 grams of fatty fish twice a week or from a supplement. And so we see a lot of individuals who are supplement sensitive. In fact, given our current clientele and the health conscious sort of bent of their mentality, these people are if you're testing with us, you're probably taking supplements. You're probably health conscious. You're probably taking some form of supplements. And what we see is actually in our current customer base, a trend for over-supplementation. We see individuals who take supplements early in the morning and they're essentially incapacitated until noon. And they have to sit down and they have the same sort of bloating, sweating, flushing, hypothermia even, and that is very characteristic, unfortunately. So we will be able to tell you if you should supplement, if you should, instead of supplementing, get that micronutrient from the food, which foods, how much, in addition to, are you over supplementing, and what should you cut out or at least cut back on. So yes, supplementation is something that we can speak very directly to, and that's because of the inherent importance of metabolites in nutrition and nutritional biochemistry.
So she also seems to be pretty interested in her health, because she's also asking, she's slow to repair or recover from exercise. Is that something that we capture in the test? I know the answer already, but.
Yeah, absolutely. So we see, for instance, even on this disease analysis report that we brought up, you see here that we have, second from the bottom, metabolites affected by exercise. Exercise, the output of metabolic output of physical activity has its own discrete signature, and it's something we can tailor to. So if you're seeing fatigue or you're slow to recover post-exercise, there are specific supplements, dietary changes, even specific changes in the exercise regimen that can be prescribed to ameliorate that.
Okay, this one is, well, let's see what your answer is. How can this test add value to the treatment protocol for cancer patients? I think you should probably have a disclaimer for answering.
Yeah, absolutely. So how can it add value? Well, like we mentioned, we can pinpoint markers that are related to cancer, report on their match. If taking our tests serially through time, then historical changes in those levels can obviously inform treatment, cease treatment, change treatment, and those clinical decisions related. I will say that metabolomics has been, if you had to pick one specialty that has had the greatest adoption of metabolomics, it's the oncologists. Given the sensitive nature of metabolites and the preclinical manifestation of metabolic signatures, oftentimes years before traditional clinical tests can adequately detect a signal of that tumor, or years before the tumor has grown large enough to be detected from a radiographic or imaging technique, metabolites are present. So oncologists have used metabolomics and their own awareness and interpretation of metabolite levels, specifically things such as alpha-ketoglutarate is a big marker of cancer metabolism. The Warburg effect is a big metabolic shift that is characteristic of generalized cancer. And so they've used metabolite measurements over time to assess the patient's prognosis and therapeutic response. Our test can essentially address inflammatory processes like we discussed that are known to cancers, but also address various other processes that are important in cancer care, such as nutrition, such as adequate exercise regimen. I mean, all of these are factored into our modeling. And so for patients, for instance, we had a patient who is on tube feeding and is bedridden. Exercise is not possible. Certain joint extension and mobility and flexibility manipulations are possible with the help of a caretaker. And that is what our algorithm and digital twinning results recommended for that individual. So in an issue where, you have a cancer patient who may be struggling with nutritional supplementary or exercise choices, this can really fine tune and enhance their care and ultimately outcome.
Perfect. Does your testing include concerns with hormonal imbalances?
Yes. So hormones obviously are proteins, and so to get a very direct view of hormones and their various levels, you would need to do some sort of proteomic level investigation, usually some clinical tests. What we are assessing with the metabolites are processes that affect and are affected by various hormones. So for instance, we can understand and get a very sensitive indicator of cortisol levels, given the metabolites that we know are associated with the HPA axis, or the hypothalamus pituitary adrenal axis, which is really this physiological response that results in high cortisol release, the stress hormone. So we can look at metabolites that are involved in the synthesis and release and metabolism of cortisol and get a very sensitive picture of, well, is too much being synthesized, is too much being released, is not enough being metabolized or taken back up into cells. That is what this very high throughput multiplex nature of our test affords. Rather than just saying your cortisol is high or low, we can say that because we're looking at production, secretion, and digestion rates of cortisol. We can infer to the level of cortisol directly, but more specifically and more importantly, we can look at, well, is there something wrong with the biosynthesis? Is there something wrong with the cellular transport? Is there something wrong with the cellular uptake of that hormone? So we don't look at hormones directly, but yet we have plans to, of course, develop a proteomics assay, but the metabolites, as we mentioned, are far more sensitive and can give us more information than just a static cortisol level or hormone level could by itself.
So this is the last question that I see.
Okay.
It's from Julie. This kind of relates to kind of detecting where inflammation is arising from. She's asking, can you tell if you have inflammation in the jaw or teeth, in the oral cavity?
Yes. So one of the metabolites that we don't have, the disease enrichment tests, as I mentioned, these are the top 25 for viewing purposes. There are like 84 or 88 different localizations that our test can significantly monitor and surveil. One of them is oral infection. So absolutely, an oral infection is one of the most overlooked sites of inflammation and can result in systemic prolonged inflammation if not adequately monitored and addressed. So 100% that is something that we can monitor. It's something that we know in the literature is a proven sort of instigator of inflammation and often overlooked, unfortunately. So it is in our panel, even though it's not in the top 25. It is in the total list, yes.
Okay, actually, I did get one more question coming from Becky. Can testing explain extreme constipation?
Come on, this is you.
Yeah,
this is right
me.
up your alley.
This is me. So, unfortunately, we're not able to capture everything that is proximal to the gastrointestinal tract with the Aristotle. So we're really trying to onboard the next evolution, I think, of Theriome, and that is the ilia. So the ilia, what it intends to do, and again, working within the systems biology framework, is profiling the microorganisms in your gut as well as the metabolism of your gut. So this is going to give a really granular indication or really granular information on what's going on with your gastrointestinal health. This includes, of course, pick your poison with gastrointestinal symptomology, particularly the lower intestines. And most people are experiencing, in the Western world anyway, they're experiencing some type of gastrointestinal symptomology, whether it be, constipation on one end or, diarrhea on the other, particularly related to transit time. So we're really looking to provide the granular type of detail and answers that, and Becky, you're not the first, and this is definitely something that's needed. And I don't think it's really addressed very well in the medical establishment or with at-home, the available at-home tests. So we're really looking forward to that. We do have a domain that focuses on gastrointestinal health with the Aristotle. Unfortunately, it's not able to get at the pinpoint type of accuracy that I think it's going to probably be needed for some of these more complex cases. Did you want to add anything on to that?
No, as Alex said, we can look at things related to, for instance, intestinal motility, which is a factor that contributes to constipation. We can look at, potential digestive imbalances regarding, with, some of the self-reported foods in your diet. It might not be really amenable to your particular digestive profile as assessed by our metabolites. So we can look at that, and if it's an issue that really pops up as being flagged and significantly impeding a certain domain, then we can offer suggestions and specific protocols to ameliorate it. But again, constipation itself, it is part of our disease signature report. Constipation, diarrhea, and various iterations of it, colitis even, are in our disease signature report. So you may have to see that pop up as significantly enriched should you order our testing. But again, we are trying to only speak to what we know is scientifically grounded and cogent and has been shown to be valid with the assessment of these metabolites. And right now, we're not quite yet to being able to say that this is the root cause of constipation and what you should do about it. But very soon, second quarter of 24, we're launching the most advanced stool test of your gut microbiota available on the market or in your doctor's office. This is really next generation sequencing and a very sophisticated, robust bioinformatics pipeline. So we'll be able to get to those conditions soon.
Good comment, actually, from Carol, not a question, and I do want to bring this up. I'm so excited about this test. I've spent upwards of $100,000 trying to get to the root cause. This test is a bargain. I'm excited and a little apprehensive. We don't blame you. However, I will say it's very simple. We don't have a test here with us to kind of show you how it works, but basically there's a small sterile lancet that's provided in each kit, and it's literally as simple as the prick of a finger or a thumb. Granted that you're sterilizing it beforehand. It's very, very simple. And as we said, our process is streamlined, our turnaround time is quick. And it seems like you've got a console as well, which is excellent. So I think you're going to be really pleased. Looks like there is one more question. I think we're going to end it on this. And this is coming back to CRP. So I've heard from some doctors, or I've heard some doctors say that high sensitive CRP is specific for the heart and cardiovascular system's inflammation. While CRP is whole body inflammation metric, and others, okay, basically saying what some parties are saying and what other parties are saying. So others are saying it's a good indication of whole body inflammation. Others say high sensitivity is better than CRP, and that there's no reason whatsoever to run CRP when you can run a high sensitive CRP.
Yeah, and
so is there any truth behind this?
Yeah, so in truth, what we discussed this just briefly earlier, that the high sensitivity format has completely obviated the need for regular CRP testing. And that the high sensitivity format is more attuned to inflammation related to the cardiovascular system and the liver. Both are true. However, it's again highly variable among individuals. So some people may have high sensitivity CRP levels that are prodromal to the disease, but don't necessarily reflect any metabolic shifts in the heart or the liver. So when we talk about CRP, it's a protein that's released by the liver, it's into the bloodstream, it causes inflammation, and it causes it's a response to infection, autoimmune disorders, etc. This high sensitivity is just a more sensitive measure of that same parameter, and it has had greater associations with risk of cardiovascular disease. So heart attacks and strokes, it has been implicated there. So it's often used to assess cardiovascular disease, but like regular CRP, the high sensitivity format can still be elevated due to sort of idiopathic inflammation, what's termed idiopathic or the cause is not known. And in high sensitivity CRP in some studies has again shown particular sensitivity for liver and cardiac diseases and disturbances. However, some studies have again just showed it to be a general response to prediction. The key is in understanding that you're not detecting a separate compound. You are just using a more sensitive platform to detect it at lower levels that otherwise would be missed with the standard testing form.
Excellent. Well, I think that pretty comprehensively answers that. I just remind everyone one more time that please take advantage of the 10% off inflammation, all caps. The consultations, I think that is the way to go if that's in your price range. We just see the best reception and compliance with that. But you know what? Any one of these categories is going to be really revealing. And we offer you to try it out. And we're excited to get your feedback. We're excited to talk with you. You choose the consultation route, and also to answer any questions that you have if you want to shoot us an email. Thank you, everybody. And of course, thank you, Dr. Jasbi, for taking us through this exciting journey with inflammation. And we hope to see you guys next time.
Thank you guys. Thank you for attending.